Hello everyone. So today we'll be talking about B cell maturation, activation and differentiation. So this is week 8's content. I am Seemanti Aditya, a Ph.D. scholar in the Department of Biotechnology at IIT Madras, and I am a teaching assistant for this course. So I take classes every Friday from 7 p.m. to 8 p.m. And so at the end of the class we'll be discussing some sample problems regarding to this topic. So okay, so let's begin. So last class we were discussing about T cell development, and so how T cell development and B cell development differs is in B cells, the cells basically it grows and develops and matures in the bone marrow itself. But in T cells, we had seen that the T cells which initially was produced in the bone marrow moved to the thymus for their maturation. So in case of B cell, both the maturation and the development take place in the bone marrow. So three stages of development of B cells are maturation, activation, and differentiation. So what do you mean by these three terms? Basically, maturation indicates when the cells are from the stem cells. So when the stem cells gets uh, differentiated to B cells, mature B cells. So that is the step which is known as maturation. So from initial stem cell stage, the hematopoietic stem cells, as it along the way as it goes on to become a B cell, that is the process which is known as maturation. What is activation is when B cells are interacting with different kind of antigens. So B cells will be interacting with different kind of antigens. So then they are basically the B cell receptor on their surface will be so this is the B cell, suppose, and B cell has a receptor on its surface. What is the B cell receptor called? So what is this B cell receptor? Anybody? We have a name for the B cell receptor. It's very common. Like with what antigen interacts? Okay, so this is basically the B cell receptor is an antibody. So on the surface of B cells we have antibody or the B cell receptor. So as the B cells are matured enough and they are released from the surface of bone marrow, they will go and interact with different antigens, and this antigen will lead to the activation of the B cells. So activation here refers to the interaction of the antigen with the perfect antibody or the perfect B cell receptor on the surface of B cells. And once the perfect antigen is fitting to the perfect antigen antibody interaction take place, then the last step comes to play, that is differentiation. So when an antigen antibody interaction is antigen antibody interaction which is perfect fit, so that there we will get the differentiation of differentiation of the B cells, that involves differentiation into memory cells, memory B cells, and plasma B cells, okay, or plasma cells. So there are as I mentioned, B cells, they are produced and mature in the bone marrow itself, and activation of mature B cells will take place in the lymph nodes and spleen. So lymph nodes and spleen, these are the lymphoid organs, and in these lymphoid organs we have several antigens coming across, and it is basically the fighting ground for all these immune responses. All your B cells and T cells will go and interact with the antigen over here in the lymph nodes and spleen. So these are the secondary lymphoid organs, and we will so activation of mature B cell, as I mentioned, was interaction with antigens. Antigen will be present in lymph nodes and spleen, and here we will the perfect antigen antibody interaction whenever it takes place. So that B cell which is having the perfect fit with the antigen, that B cell will be differentiated. So your B cell will be differentiated into memory B cell and plasma cells. And also there will be clonal expansion. So like particularly one B cell will be growing into several B cells. So there will be clonal expansion of those B cells, and from clonal expansion you will get the two different populations, that is memory and plasma. So that is what is the development of B cells in a broad prospect. Like the three stages are maturation, activation, and differentiation. Now, if we look into the step of maturation, so what happens is, so this is the bone marrow. So this picture here represents bone marrow, and from bone marrow we have the hematopoietic stem cells. So these are the hematopoietic stem cells, and the hematopoietic stem cells basically will be growing and differentiating into different stages. So first stage over here, as it is mentioned, is early pro B cells. So before that we also have a progenitor cell. So these are progenitor cells are present. So as soon as the hematopoietic stem cells will be differentiating into progenitor cells, and progenitor cells from there will be differentiated into the early pro B cells, then the late pro B cells or pre B cells, and then comes the third as the immature B cells. So this is one, hematopoietic stem cells, then comes the progenitor cells, then comes the early pro B cells, then comes the pro B or pre B. This is basically late pro B or pre B cells. This is the third stage. Then comes the immature B cells. So immature B cells from here is transition to your spleen or lymph node, where it will actually get matured. So here it is the maturation and the activation will take place. So this is the fifth step. Right. So what happens is the hematopoietic stem cells get differentiated into progenitor cells. Then the progenitor cells with expression of different kind of receptors on their surface, and due to the expression of different cytokines, these progenitor cells will develop into pro B cells. And again, due to the expression of different cytokines and receptors, it will get converted to pre B cell or pro B cell, late pro B cells, and finally an immature B cell, and then it will get finally converted to a mature B cell. So what are these cytokines and what are these receptors that are distinguishing each of these stages from the previous one? So basically in hematopoietic stem cell, it does not have any kind of differentiating factor. These cells can basically differentiate into any kind of cells in your blood. It can be erythrocytes, monocytes, leukocytes, whatever. So those do not have any kind of differentiating factors, your hematopoietic stem cells. But once it is coming to a progenitor cells will start expressing certain kind of receptors. So those receptors are like FLT3. So this is a receptor which is expressed on the surface of progenitor cells, and this is a tyrosine kinase receptor. So all this tyrosine kinase receptor, or say the EGFR receptor, these are basically responsible for signaling pathways, and this signaling is very important in terms of cell biology, because this signaling will lead to the differentiation of cells, will lead to the differentiation of tissues. So basically the progenitor cells expressing FLT3 allows the progenitor cells to get differentiated from the hematopoietic stem cells, and it can this has a criteria, this has a probability that it can go into a B cell, or it can go into any kind of erythrocytes or say monocytes. So once the progenitor cells is reached, so it's reached, and then there will be an expression of another receptor, which is IL-7R. So IL-7R is an interleukin receptor. So these receptors basically are responsible for distinguishing this pro B cells to be committed towards the B cell lineage. So till here, it's not still sure whether the cell is going to be a B cell or a T cell or a other kind of cell. So once the progenitor cells expressing FLT3, this again mediates certain kind of signaling with tyrosine. It is a tyrosine kinase receptor. So it will mediate certain kind of signaling, and those signaling will again lead to the production of this particular stage of B cell, and this particular stage of B cell will express the receptors like interleukin 7R, and this is responsible for committing this cell to the B cell lineage. So this B cell lineage commitment is achieved over here. So now the cell is sure enough that it is going to become a B cell. So here in early B cell, what happens is the DJ heavy chain recombination takes place, and there can be a start of VDJ heavy chain recombination as well. So we have talked about the VDJ recombination and the DJ recombination. So again, briefly, what is VDJ and what is DJ recombination? So heavy chain has different segments. So heavy chain has different kind of segments like mu segment, and then delta, and then gamma, etc. So in each of these segments has several parts, one, mu2, etc. So each of these parts will have, like, suppose this is the heavy chain region. Your heavy chain is responsible for your antibody subtype, right? So heavy chain will be responsible for distinguishing whether your antibody is IgM or whether it is IgD or whether it is IgE, based on whatever segment it is there. So if it is mu, it is IgM. If it is delta, it is IgD. If it is gamma, it is IgG. So likewise. Now, so in heavy chain recombination, what happens is in this B cell, so your B cell is having your antibody with the heavy chain, just the heavy chain, and in your heavy chain, it could be an the variable region of your heavy chain has the VDJ part, and the constant region will have the VDJ part. So there are two parts in the heavy chain as well, right? The variable region and the constant region. So these parts get recombined to form different kind of antibodies with different specificities to antigen. So those process, so VDJ recombination involves like a mix and match of the heavy chain segments in order to get a variety of heavy chain domains of antibody that could interact with different antigen. So your early pro B cell, here your heavy chain recombination will start in the early pro B cell. Completed, and your light chain recombination has started. So your light chain will start forming over here. So your light chain recombination will your clonal expansion starts. So what is clonal expansion? So any kind of cell, any kind of B cell that is expressing any your antibody of interest. So this is one important thing of your B cell. So this is just IgM type. Why IgM type? If you are looking into the segment which was again taught earlier classes, the mu segment comes, the mu segment of your heavy chain comes the first, and therefore during recombination, during this VDJ recombination, the mu segment is taken at the first place, and therefore the IgM type of antibody is produced during maturation. So yeah, in this stages, in early pro B cell and late pro B cell, your heavy chain and light chain recombination will take place, and once it is done, you have an IgM type of antibody expressing on the surface of your B cell. So this is the blue colored part you are seeing over here. These are the IgM type of antibodies. And so clonal expansion means that one cell that is expressing your IgM type of antibody after your recombination. So there are several types of recombination that is taking place. Say B cell 1, B cell 2, B cell 3, Okay, so this is on the basis of what kind of antibody rearrangement has taken place. So there will be different kind of antibody rearrangement that is specific to different antigen. So whenever this B cell 1 is expanding into a population, that is the clonal expansion. Again, B cell 2 will also expand into a certain population, that is clonal expansion 2. Also likewise. So clonal expansion means one cell is getting divided into a group of cells. So yeah, so after this stage, what happens is once the cells which are expressing your IgM type, your fully recombined IgM type of antibody, so those IgM type of antibody will be interacting with endogenous antigen. So what are endogenous antigen? So antigens in the bone marrow, like your bone marrow has certain cells which are known as, say, stromal cells. So stromal cells are there for a reason. Like what reason? So stromal cells will express certain antigen on its surface, that is self antigen, right? So your antigen which is present in the stromal cells, that is your self antigen, and B cells which are recognizing this self antigen should be ideally eliminated, because if it is interacting with your self antigens, then there is a chance that in the future also it can interact with your self antigen and cause autoimmune disorders. So we do not want that. So here what happens is a negative selection, where your B cells expressing the IgM type of antibody interacting with your endogenous antigen, like the self antigen which is expressed on the surface of stromal cells in the bone marrow, will be eliminated. Now, during this elimination, there can be two steps that could occur. Either it is totally deleted, that cell is totally deleted and it dies by apoptosis, or again this cell will go in another round of this VDJ recombination to edit its receptor, and after that set of recombination, if it finds a new set of receptor that is not interacting with your self antigen, it will again proceed with the further step of maturation. Otherwise, it is deleted. Right. So what happens is now your cells which are interacting with your self antigen is deleted. That is known as negative selection. And finally, your cells which are not interacting, that will be proceeding towards your periphery of the bone marrow. So this is towards your central part of your bone marrow, and as it is getting mature, it is towards the periphery of the bone marrow. And in the periphery of the bone marrow, once it is not interacting with the self antigen, it goes and it will be transitioned, or it will be transported to your lymph nodes. So spleen is a lymph node. Spleen is again a lymph node in your body. So where maximum amount of antigen antibody interaction can take place. It's a very big lymph node. So your immature B cells, at this stage it is known as immature B cells. Immature B cells. So this is immature B cells. Once it is coming towards your spleen or lymph node, here what happens, it is coming in contact with antigens which are not your self antigen, with basically your antigen from exogenous sources. So exogenous antigen exposure. So exogenous antigen exposure will lead to the development of another kind of antibody type in your cells, in your B cells, that is the IgD type. So here the exogenous antigen exposure will lead to the positive selection of your B cells. So cells which are interacting with those kind of antigen which are in the exogenous sources like bacteria or virus or whatever your parasite. So those cells will be selected positively, and again it will undergo a process of clonal selection, and finally it will get converted to a mature B cell. So what is the characteristic of mature B cell? So mature B cell needs to express both IgM and IgD type of antibody. Whereas in immature B cell, we will see that there is expression of only IgM. Immature B cells has expression of only IgM. Okay. So this is the difference between immature and mature B cells. And also mature B cells, it is coming in contact with exogenous antigen, whereas immature B cells, they are coming in contact with self antigen. And at that stage, all the cells which are interacting with self antigen will be deleted. So this is how the entire development of B cells take place in the bone marrow and in the lymph node. Now next we will see what are the expression of different cell surface markers on the hematopoietic stem cells and the lymphoid progenitor cells. That is basically giving the identity of this particular cell and how it is differentiating that particular stage from different stages. So there are different kind of cell surface markers or receptors. So these are c-KIT, SCA1, FLT3, CD34, IL7R, RAG1, 2, and TDT. So during hematopoietic stem cell development, so hematopoietic stem cells, it can express this c-KIT and SCA1 kind of receptor, and it is not having any kind of commitment towards any kind of cell type. So it can again differentiate into its same type. Hematopoietic stem cells can differentiate into its own. So here the arrow shows that hematopoietic stem cells can divide and form its again a stem cell, or it can get differentiated into a multipotent progenitor cell. And from multipotent progenitor cell, we can get two types of cells, that is erythrocyte and megakaryocyte. And multipotent progenitor cells also expresses c-KIT, SCA1, and along with that another receptor which is CD34. So you see there are certain changes in the receptor expression in different stages. So that is basically distinguishing a particular stage from the other stage. So c-KIT, SCA1, and CD34 is expressed in multipotent progenitor. Again, this will get differentiated into a lymphocyte progenitor cell. So lymphocyte progenitor cell can differentiate and can give rise to monocyte, and this expresses c-KIT, SCA1, FLT3, and not CD34. So why are these receptors expressed? So these receptors are basically expressed in order to interact with certain cytokines, or interact with interleukins or interferons. So these receptors can interact with different cytokines or interleukins or interferons. So whenever there is different set of cytokines which is produced, it will give rise to that particular type of cell. So if it is expressing, say, c-KIT can interact with some kind of cytokines that will give rise to either megakaryocyte. If it is interacting with other kind of cytokines, it will give rise to erythrocyte. So that is how it works. So these receptors are basically giving identity and also helping in interacting with different cytokines and interleukins and interferons for differentiating the particular cell type. Right. So so from hematopoietic stem cell, it will come to a multipotent progenitor cell, which is known as MPP. Then it will come to a lymphocyte progenitor cell, which is known as LMPP. Then it is a early lymphoid progenitor cell, which gives rise to T cell progenitor, and from here we'll get our T cells develop from here. And from early lymphoid progenitor cell, we get the expression of common lymphoid precursor. So here you see that this is responsible for the production of pre-pro P cell, as well as dendritic cell, as well as natural killer cell and T cell progenitor cells. So common lymphoid precursor can give rise to either type of cell, B cell, NK cell, DC cell, or T cell progenitor. But if it is mainly expressing IL7R or the interleukin 7 receptor, so when the cells are, common lymphoid precursor cell, when it gets differentiated to pre-pro B cell, it is basically expressing this IL7 receptor in a higher amount. So IL7 receptor in a higher amount leads to the commitment of B cell lineage. So other receptors, if it is expressed in maybe this IL-7 receptor is expressed here as well, here as well, here as well, but it is expressed in a higher amount in case of the cell which is committed towards the B cell lineage. So apart from all other receptors, the IL-7 receptor is very important in terms of commitment of the cells towards the B cells. So once it is expressing the IL-7R, we are having the pre-pro B cell, and this is going to get converted to pro B cell, and that is going to get converted into the mature B cell, and finally the mature B cells. So that is the development of the B cells. Now, how the maturation and clonal selection of B cells take place. So again, just a brush up. So stem cells, this is present in your bone marrow, and your stem cell will have no kind of B cell receptor on its surface. There will be certain gene arrangements of this B cell receptor. So gene arrangement of B cell receptor, which is also an antibody molecule. So yeah, so gene arrangement of B cell receptor will give rise to the antibody or the B cell receptors on expressed on the surface of your immature B cells. So immature B cells has only IgM type of antibody. See, there is only blue colored IgM type of antibody in immature B cells. So these are clone number 1, clone number 2, clone number 3, clone number 4, based on the different kind of antigen specificity. So clone number 1 is having antigen specificity to antigen 1. It is having antigen specificity to antigen 2. It is having clone number 3 is having antigen specificity to antigen 3. Clone number 4 is having antigen specificity to antigen 4. Now see, in different kind of clone, so again the immature B cell will be going towards your node, your lymph nodes, and here it will come across no, not before that, it will interact with your bone marrow cells as well, the stromal cells. So IgM type of antibody which is interacting with any self antigen, that will get deleted. So see, the clone number 4 here shows interaction with antigen 4, and that happens to be a self antigen. So that clone gets deleted. So clone gets deleted, and it will not proceed towards maturation. But if it is not interacting with your self antigen, then it will proceed towards maturation. So maturation will involve the development of both the types of antibody on its surface, say IgD type as well as IgM type. Now what happens is, your mature B cell will come in contact with several antigen on the surface of your lymphoid tissues. So your lymphoid tissues are your spleen, your lymph nodes. So when it comes in contact with your exogenous antigen, so this is your exogenous antigen, the antigen which is coming from your external surface, and this is your your antigen which is in your body. So here there is a negative selection will take place, here there is positive selection that takes place. So this is negative selection, this step is for negative selection, and this step is for positive selection. So basically, after your negative selection, you have 3 clones that are not interacting with your self antigen, which is expressed on the surface of stromal cells. So your 3 clones will expand and show clonal expansion. And out of your 3 clones, when these 3 B cells come in contact with exogenous antigen in your lymphoid tissue, that antigen which is perfectly fitting with your antibodies expressed on the surface, say from clone number 1, 2, and 3, the antibodies which are expressed on the surface of clone number 2 is perfectly interacting with antigen 2. So this particular clone will be expanded into a population. Rest 2 mature B cells will remain in the circulation, and if it does not find an antibody, if it does not find an antigen of interest within some duration of days, it will die. So, so your antibody, which is interacting with your the proper antigen, that will get expanded and differentiated. So your clone 2 gets differentiated into your set of memory cells, and set of plasma cells. So there is different kind of expansion. So there is a big expansion where it is antigen dependent proliferation and differentiation into plasma and memory B cells. So what is memory B cells? So memory B cells will have this embedded antigen on its surface and can detect antigen that will probably infect you again 10 to 20 years later. Whereas plasma B cells, they have cells that can secrete the antibody into your blood, and it is not going to it is not going to stay for a longer period of time. So this plasma cells will do its job. It will kill the cells, it will show opsonization, or it will neutralize your antigen by releasing the antibody in the blood, and finally it will it will clear your body of infection. And whereas memory cell will remain in your blood circulation for a long period of time. So this is how clonal selection takes place. So again, a brief overview. Your stem cells gets developed into different progenitor cells. Progenitor cells, there will be gene rearrangement of B cell receptors that will result in different antibodies or different B cell receptor with different antigen specificity. And so we are having 4 different clones. Out of 4 different clones, one clone that is not interacting with your that is interacting with your self antigen gets eliminated. The rest 3 clones will proceed further in the steps. So out of the rest 3 clones that are matured, the clone that is interacting with the antigen of interest gets the clone that interacts with the antigen of interest will get clonally selected and mature into your memory B cell and plasma cells. So B cell activation can be 2 types. Basically, that can be either thymus dependent, or it can be thymus independent. So what do as the name suggests, that B cell activation, whether it can be thymus dependent or thymus independent. So if it is interacting with T cells in the thymus region, then the B cell activation becomes thymus dependent. If it is not, if the B cell activation is independent of interaction with T cells, then it is thymus independent. So 2 pathways, basically thymus dependent and thymus independent. So thymus dependent pathway will involve interaction of B cell with T helper cells, and this interaction will lead to the B cell activation. Whereas thymus independent pathway is based on the B cell activation due to downstream signaling responses on account of B cell interaction with LPS or bacterial DNA directly, and no scene of B cell and T helper cell interaction. So look at the first figure. Here we have thymus dependent antigen. So this is our B cell. Our B cell is interacting with this antibody. These are B cell receptors, antibodies interacting with certain antigens, and this B cell will engulf the it will interact with the antigen, process it, and then express it on the surface of an MHC molecule that will be presenting this antigen to the T helper cells. So T helper cells has T cell receptor recognizing antigen which is presented by MHC molecule. And so once this interaction takes place, there will be a second interaction, that is CD40 and CD40L. So CD40 ligand is present on the surface of T helper cells. That also interacts with a CD40 receptor that is present on the surface of the B cell. So both interaction will lead to the activation of B cells. Basically, the B cell that is interacting with this antigen will get activated and will show clonal expansion. And this activation will lead to the production of cytokines from T helper cells. So what are cytokines? Again, cytokines, as I said, that it can be different kind of so the T helper cells will secrete different kinds of cytokines that is like IL-2 or IL-4, those kinds, and it is going to interact with the receptors present on the surface of B cell and help in the proliferation and differentiation of B cell. So there are 3 interactions which is important for the thymus dependent B cell activation. The first interaction is the antigen antibody interaction in the B cell. The second interaction is the CD40 ligand and CD40 receptor present on the surface of B cell and T cell. The third, as well as the the interaction between the MHC receptor, MHC presenting the antigen to the T cell receptor, and the third kind of interaction is the cytokines which are released from T helper cells. So this is how the activation of B cell will take place through all these three signaling pathways. In thymus independent B cell activation, that is again divided into two types. So that can be thymus independent type 1 or thymus independent type 2. So in case of thymus independent type 1, mainly the LPS, so you have heard of LPS, the lipopolysaccharide layer of a gram-negative bacteria, as well as small bacterial DNA like such. So those LPS and bacterial DNA, these are highly immunogenic, and this do not require any T helper cell activation. So LPS can directly interact with the TLR or toll-like receptors. These are innate immune receptors which are present on the surface of B cell, and it can activate a signaling pathway. So this signaling pathway is basically mediated by this toll-like receptors domain, as well as the antigen which is interacting with the antibody. So here, after interaction with the toll-like receptor and the antibody, it will mediate a signaling response through another cytosolic domain which is known as ITAM. So what is ITAM? This ITAM is immune receptor tyrosine based activation motif. So ITAM, I can write, I'll just write. So this is ITAM. So ITAM is going to, this is immunoreceptor tyrosine activated motifs. So what is ITAM doing? Once your antigen is interacting with your toll-like receptors, ITAM will go and activate the different kind of signal like transcription factors. So there can be activation of different okay, so my pen is not working. So again, so the item after interacting with the antigen after interacting with toll-like receptors will signal the item molecule. This is the cytosolic domain of the toll-like receptor, and this item will send a signaling response through different chemokines and cytokines to the transcription factors. So transcription factors will get activated, and that transcription factors will lead to the differentiation and proliferation of this particular B cells. This is also the mechanism. Same mechanism goes for the thymus independent type 2. Only the difference is in type 1 antigen and type 2 antigen, what kind of antigen they are recognizing. So type 1 antigen is basically the LPS and bacterial DNA, and type 2 antigen is mostly the repetitive structures. So what are the repetitive structures? So repetitive structures could be some your repetitive DNA or repetitive structures which are having repetitive amino acid sequences, there are hairpin loops, or maybe the RNA molecules which are having some antigenic property. So like hairpin RNAs, like that. So the mechanism of type 2 antigen is it is binding to the complement determining like the complement protein C3d, and this complement protein will show a cross linking with the antigen, like the antigen it is having repetitive structure, right? So the antigen will be interacting with your antibody as well as the complement protein, and will form a cross-linked structure, and this cross-linked structure will send the signaling to the transcription factors for activating this B cell. So this signaling is again similar to this item signaling, where it is activating several transcription factors, and it activates several genes for differentiating the B cells. So this is how the different kind of activation of B cell takes place. So Thymus dependent activation helps in the activation of B cell due to the interaction with Th cell, whereas in Thymus independent there is no requirement of T cell interaction with B cells. Just the antigen antibody interaction and the antigen B cell interaction is enough for proliferation and activation of B cell. Also in type 2, there could be a requirement that certain cells like dendritic cells is required for kind of activation, but not T helper cells. Now there is the steps of B cell activation and differentiation. Again, how it takes place. So this is a conventional naive B cell. Naive B cell means a cell which has not been exposed to any kind of exogenous antigen. So once the naive B cell is exposed to an antigen, so this is a thymus dependent antigen pathway. So either thymus dependent antigen pathway will directly lead to the clonal expansion and clonal expansion of B cell that will get matured enough to produce the antibodies against that antigen, or it will again go through the germinal center and increase affinity of the antibody receptor will be obtained. So basically, if a B cell is exposed to a thymus dependent antigen, then it will directly lead to the clonal expansion of this B cell, or it might lead to a better improved variety of this B cell that is having better affinity to the antigen. So T dependent antigen activation will lead to the maturation of will lead to the activation of B cell in the germinal center. Now, what is germinal center? So germinal centers are present in the lymph nodes, and it is responsible for B cell activation and maturation. And mostly the population in the germinal center is of B cells. So once the naive B cell is exposed to a TD antigen or thymus dependent antigen, it will enter the germinal center and it will proliferate, show the class switching recombination or the SHM. Class switching recombination, or maybe some sort of anti like the so SHM was okay. I'll just get back to this SHM. Okay, anyway, so proliferation, SHM and CSR. So CSR means class switching recombination, and your cells which are expressing particular antibody against your antigen, if it is showing lower affinity, it will die by apoptosis, and if it is showing a medium affinity, then it will go and bind to your follicular dendritic cells, which is present on the surface of the germinal center, and also it will interact with the T helper cells in the germinal center, and this interaction will increase the affinity of your antibody towards your antigen of interest, and that is going to lead the expansion of this particular B cell with a better improved affinity. So affinity is going to be improved because there can be some somatic hyper. So SHM stands for somatic hypermutation. Yeah, so somatic hypermutation and class switching recombination is going to lead a better variety of your B cell with a better antibody that can interact with your antigen. So we know that somatic hypermutation takes place only in B cell. What is somatic hypermutation? Just a brief overview. So somatic hypermutation will lead to a mutation in the variable region of your antigen antibody. So if your mutation in a variable region of antibody leads to a better variety of an antibody that could interact with your antigen with a better affinity, so that antibody is selected, while the less affinity interactions will be deleted, or those cells will die by apoptosis. So this is what is happening in the germinal center. So basically the entering of B cells in the germinal center leads to the increased affinity of your B cell towards B cell expressing the antibody towards your antigen. And if it is not entering the germinal center, now what on what basis does it enter the germinal center, or whether it does not enter the germinal center and just gets proliferated into your clone of cells which is expressing the same type of antibody that is interacting with antigen, that is not yet known. And we like basically different booster doses are given for this, so that your B cell will enter this germinal center and go through a better way of maturation and activation, so that your antibodies are very like perfect for your antigen and can neutralize your antigen at the best possible way. So your naive B cells, which are proliferating and showing somatic hypermutation and class switching, so this is which are having low affinity and very autoreactive species, or species which are interacting with self antigens. So those those antibodies are basically eliminated and by apoptosis, whereas the ones which is not which is showing a medium or moderate kind of interaction will lead towards a better mutation in the variable region of your antibody, and those antibody will show class switching as well as differentiating into memory cells as well as plasma cells. So basically this particular B cell, which is showing improved affinity, and due to the somatic hypermutation, it will proliferate into a plasma cell and memory B cell, and as well as class switched memory B cell. What is class switching? So class switching involves switching between different subtypes of antibody, that is either isotypes of antibody, that is between IgM or IgD, IgG, IgE, etc. So this particular B cell will then further get differentiated. So this is what is differentiation. So first is maturation, then it is activation, then is differentiation. So these are three stages of your B cell development. So maturation takes place in the bone marrow as well as lymph node, where it gets where the mature B cell expresses both IgM and IgD type of antibody. After it is mature enough, it is going towards the interaction with germinal centers in the lymph nodes, where it is coming across the different exogenous antigen. There it will go and show somatic hypermutation, and the perfect antibody which is having the perfect and fit with the antigen will show clonal expansion and get differentiated into the plasma cells and memory B cells, as well as class switching will take place, where your IgM type of antibody or IgD type of antibody, which was initially produced, will get switched into different types like IgE or IgG or IgA, etc. So antibody class switching, again, what is antibody class switching? So this is the VDJ part of your heavy chain, and your heavy chain has this different segments. So C mu, C delta, C gamma 3, 1, 2b, etc. So what is C mu representing? C mu is the IgM type of antibody. C delta is the IgD type of antibody. C gamma is for IgG, and C E is for IgE, and C alpha is for IgA. So this is just a representation of how IgM type of antibody will get converted to IgE type of antibody. So there are certain regions in your segments of your initially during development. This is the genetic level. So in your gene, in your antibody gene, there is certain regions which is known as the switch region, or designated as S. So this switch region gets cleaved or gets a double-stranded break by this enzyme, which is known as AID. AID stands for activation induced cytidine deaminase. So this is AID stands for activation induced cytidine deaminase. What it does is it deaminates all the cytidine residues present in this S region, and that will result in the double-stranded breaks. So these double-stranded breaks will then result in the this double stranded breaks will come towards each other, and the DNA repair mechanism will come into play. And when the DNA repair mechanism comes into play to repair the double stranded breaks, this particular region will get cut away from the your gene and will get removed, whereas the heavy chain will express only the VDJ part along with this the end part of your gene, that is CE. So it is having both CE and C alpha, but the first gene, which is CE, that is the type of antibody, that is the isotype of antibody that is going to be produced. So this is an IgE type of antibody. So initially, when your heavy chain is expressing IgM type of antibody, after your class switching, it can get converted into any type based on where your AID is cleaving. So if your AID is cleaving between here and here, so this portion will be removed and you will get an IgG type of antibody. Whereas if it is cutting between here and here, no, if it is cutting between here, this S region and this S region, so you are going to get an IgA type of antibody. So that is how your antibody class switching takes place. And there is one signaling mechanism which I thought that maybe I'll just discuss in brief. So I was talking about how IL-7 is important for B cell development, right? So IL-7 receptor is responsible for commitment towards B cell lineage. Now, as what does IL-7 do? So IL-7 is a very important interleukin, and it is responsible for activating STAT5. So STAT5 is a transcription factor, and it's a very important transcription factor where it can interact with JAK. You might have heard about JAK-STAT pathway, not signaling pathway. So these pathways are very important in cell differentiation, in cancer signaling as well. So here in our interest is IL-7 will interact with these IL-7 receptors which are expressed on the surface of your B cells, and that will result in the activation of STAT5. So STAT5 gets dimerized, and that will result in the activation of several other transcription factors like E2A, EBF1, PAX5, etc., and also NMYC and CMYC. So these transcription factors are associated with B cell proliferation, DJ recombination in the heavy chain, and VDJ recombination in the heavy chain, as well as the transcription of B cell specification and commitment survival. So IL-7 receptor binding to IL-7 binding to IL-7 receptor is important in the B cell activation, proliferation, as well as the maturation. So that is all for today's lecture. Now we will be solving some questions. So you can basically answer this. This is what is energy. So energy I have not discussed, but it was there in the lecture of your the NPTEL video lectures. So if you have seen, otherwise I can explain once. I'll just see if anybody knows what is this. For energy, so yeah, so if B cell is interacting with any self antigen which is soluble, so it will lead to unresponsiveness of this B cell. So if the B cell is not responding, your B cell is interacting with your self antigen, but it is not responding to it. It is not going to get activated or matured enough. So that is known as anergy. So anergy, the term leads to those B cells which are unresponsive to activation or maturation because it is interacting with self antigen, self soluble antigen with very, very high affinity. So the answer is A. So how is class switching of B cells brought about? Right. So it's option B, cytokines. So effector functions of T helper cells will include production of so T helper cells basically. So T helper cells are, not only just activating just the macrophages. It has other roles because T helper cells are releasing certain cytokines, and it is also responsible for the production of this. This cytokines will, if you are producing any cytokines, it is also going to attract this macrophages to the site of infection, as well as this will activate this macrophages. And what are monocytes? Actually, monocytes are the activated form of macrophages. Those are only monocytes. So it is also resulting in the production of monocytes. Monocytes are nothing but the activated form of macrophages or differentiated form of macrophages. So answer is not just A, it is all of the above. So yeah, so T helper cells, why? Because T helper cells, again, it releases cytokines, and cytokines is resulting in activation of macrophages. Activation of macrophages leads to the production of monocytes, because activated macrophages are called monocytes, and those cytokines are going to attract macrophages to the site of infection. So all of the above is the answer. Okay, not just option A. So what is this? So yeah, so the answer is option number A. LPS can lead to the activation of B cell by thymus independent. So LPS, bacterial DNA, and repetitive structures, these are leading to B cell activation by thymus independent process, and all those viral antigens, as well as these cancer antigens, and some other whole bigger antigens which are present, like particles, etc. Those are by thymus dependent process. So LPS is by thymus independent. So the answer is answer number A. This is the last question for today. Right. So the correct answer is option number A. As I discussed, so ITEM is responsible for the signaling mechanism in the thymus independent pathway. ITEM is going to activate several transcription factors. As soon as an antigen is interacting with your antibody, the Ig alpha and Ig beta part, or the cytosolic part, is known as the ITEM part, and this ITEM part is known as this ITEM part is going to activate several transcription factors, and that is leading to the activation of B cells. So that is all for today. Do you have any other questions? Okay, so if nobody has any other questions, then we will. Okay, please share the link of the 7th edition. 1 second, let me search. Okay, so that's all. Thank you, everyone. We'll meet again in the next Friday. If you have any questions, then we can discuss it next day again. So, okay, bye bye. Have a nice weekend.